Good morning to one and all. I am going to continue the lesson 4 next topic, screening for recombinants. So what is screening? So after the competent of host or introduction of the recombinant DNA into a suitable host cell means it is essential to identify those cells which have received the recombinant DNA molecule. So that process is known as a screening. So in that process, the vector or foreign DNA present in a recombinant cells that express the characteristics in the host, while non-recombinant do not express their character or traits. For this, we can use the following methods. One of the important method is a blue-white selection method. In other way, we can told as a insertional inactivation method. So, insertional means foreign gene insert panni, we can inactivate the gene in a vector. So, that is a insertional inactivation. It is one of the powerful method for screening of the recombinant plasmid. That is a recombinant DNA we can easily identified by this method. In this method, one of the main gene, reporter gene lag-EZ is present in the vector, and that gene encodes the enzyme beta-galactosidase. The beta-galactosidase, what's the function of the beta-galactosidase enzyme means? Which breaks the synthetic substrate in a culture medium. That synthetic substrate is a X-gall. So in a chemical way, we can told as a 5-bromo-4-chloro-indolyl beta-D galactopyranoside. So this is the synthetic substrate enzyme. This enzyme is break in the culture medium by the beta-galactosidase enzyme. This beta-galactosidase enzyme is encoded by the lag-EZ gene. Suppose we can introduce the foreign DNA into a lag-EZ gene means that gene will be inactivated by the insertion of the foreign DNA. So, what is the function of this X-gal in a culture medium means there is the production of the insoluble blue color in the culture medium. The inactivation of the lag-EZ gene means there is no the production of the blue color in the culture medium. So the color may be white in color. So the white color colonies represents the recombinant DNA form. Y means the foreign DNA is insert into the lag-EZ gene. So the foreign DNA is insert into the lag-EZ gene, and that means vector, and the vector is present in the host cell. So it denotes the recombinant DNA form. Suppose the blue color is present in the colony means in the culture medium means that represents the non-recombinant. Okay. So on the basis of the color, we can easily identify whether the colonies are blue or white. The blue means there is no the insertion of the foreign DNA. That is the non-recombinant. Suppose the white in color means there is the recombinant DNA easily insert into the host cell. We can easily identify and also selected the colonies easily, recombinant DNA. So here is the diagram of blue-white colony selection. So there is a vector. So PUC 18 is a vector, expression vector, which has the lag-EZ gene promoter. So ampicillin resistant characters. So here we can foreign DNA is insert means in the culture medium they produce the blue and white color. So blue color means there is no the recombinant, non-recombinant one. White color means there is the recombinant one. Okay. So we can easily select out the recombinant and regenerate as a plant easily. So this is the one of the selection method. So this is one of the technique of the screening. Next one is the antibiotic resistant markers. Antibiotic resistant marker is a gene that produces the concerned protein that provides the cells with resistance to an antibiotic. We already know the selectable marker. Okay. So in this way we can easily identify recombinants will grow in a medium and encoding the resistant capacity like ampicillin, chloramphenicol, tetracycline and kanamycin also. So while others may not be able to grow in this medium. So culture medium, we can give the culture medium and has the capacity of the resistant power, means we can easily identify the colonies and also regenerate the colonies as a plant. So one of the main character is the antibiotic resistant markers. It is also responsible to identify the selection of the colonies. And next one is the replica plating technique. This is also one of the technique. In this technique, we can easily identify the pattern of colonies growing on a culture plate, and that colonies easily copied into the another one plate. Why means we can easily lift the same colonies and also identify their relative positions in the virginal plate. But here in the second plate or copied plate, we can change the medium which is not in the similar of the culture plate, that is a master plate. Okay, so we can easily differ the second culture medium plate and also antibiotic resistant characteristics. Then only we can easily identify the transformed cells, and also that transformed cells also we can selected and regenerate as a plant. So that is also one of the selection method, easily identified the transformed cells by changing their culture medium and also antibiotic resistant characteristics. Here we can use the sterile filter paper to copy the colonies from the culture plate to the second plate. So this one is a diagrammatic of the replica plating technique. Then only we can easily understand. So this one is a master plate. So from the master plate or culture plate, here the velvet sheet and also the sterile filter paper is used to copy the colonies. So here we can change the culture medium. Okay, so this one is the ampicillin sensitive colonies, means so lack ampicillins kudukumbodhu the colonies the sensitivity we can change and also incubate the culture medium in the second plate. So resistant capacity, and that means antibiotic resistant capacity and also culture medium we can change from the master plate to the second plate. Then only we can easily identify the transformed cells and also that cells also selected and regenerate as a plant cells. Okay, thank you.